So thank you very, very much for the invitation to join your annual meeting and the opportunity to share some of our latest research with you today. The title of my talk is Genetics of Retinal Dystrophies and Patient Stem Cell Models to Test Potential Therapies. And as I'm sure you're all aware, there's huge phenotypic heterogeneity in the inherited retinal dystrophies. And they're categorized by their age of onset, the severity of the condition, and the pattern of visual loss. And there are some examples on this slide here, such as Leber's congenital amaurosis, which is an early onset condition affecting both the rod photoreceptors and the cone photoreceptors. And then we have conditions like retinitis pigmentosa, which are predominantly rods photoreceptors, but then can affect cone photoreceptors also later in the condition. And we have cone rod dystrophies. So there's a whole variety of conditions that are defined by which cell types they affect primarily. And this may also include other cells in the retina, such as the retinal pigment epithelial layer. So as well as the enormous phenotypic heterogeneity that we have for these conditions, we also have enormous genetic heterogeneity. And this really is a reflection of the complex biology and function of the retina and how we perceive light and perceive our visual world. And this graph really just illustrates the progress we've made over the last few decades. So you can see that in 1990, the first gene was discovered around this period, and that was the rhodopsin gene causing retinitis pigmentosa. And in the old days, and I remember those because I'm older than I look, it was difficult to find these genes, and we were really constrained by the techniques we had. You know, PCR was a technique that revolutionized what we could do in those early days of gene discovery for inherited retinal conditions. And then around 2003, the human genome sequence was completed. That was an enormous task that we were part of. We were focusing on cloning and sequencing the X chromosome in those days, because that was the focus of my research. And then, as you can see, in 2009, 2010, 2011, and 12, there's been another revolution in the technology that we use, and this is called next generation sequencing. And it started with exome sequencing, and now we've moved on to genome sequencing. And I'll describe some of those briefly to you as we go through the talk today. But this has absolutely revolutionized the way we can do our genetic research and make genetic discoveries. So we've gone from knowing no genes that cause these conditions to the current situation, which is we now know there's of 271 genes involved in this whole plethora of different types of retinal dystrophy. So I did say that the genetic code, you can think of like units that come into code for a protein. So I just wanted to give you a little idea of what we mean by that and how genetic variations can disrupt that genetic code. So here's an example. If we think of a gene as a sentence, and it's made up entirely of 3-letter words, because that is the genetic code that codes for a protein. Every 3 letters makes a slightly different amino acid that then makes your protein that goes into your cell. So if we think of this sentence, the sun was hot, as in the sun in the sky. If we change one letter, let's say that letter is the U in sun. Let's change that letter to an O. So we have a 1-letter change in your DNA, in your genetic code, and it now codes for the sentence, the sun was hot. So it's still a word, but it's the wrong word. So that's an example of the type of disruption to the genetic code that we're looking for in the DNA sequence. Other things that can happen is we could lose 1 letter out of these 3-letter words. So let's say we lose the U in sun. Now you can see that after there is no sentence. It doesn't make any sense at all. Similarly, we can add a letter. So if we put an H in, for example, after the S, you can see that after the sentence doesn't make any sense at all. And the other thing that can happen is we can change a letter, and then what happens is the genetic code reads it as a full stop, like a full stop in a sentence. So that's the final example, which is the full stop. So you have no sentence at all. Now, these are the relatively easy to understand how you can change one letter in the human genome, and then it is detrimental to the sentence or the gene code. And this is what we know most about in terms of genetic variation in our human genome. So we've made huge progress with this next generation sequencing approach to understanding genetic variation. And it needs really big computers and the power to process all the data. But as I said, the challenge is, which variant is it in the human genome that may be detrimental to the function of the retina? So as I mentioned before, there's about 25,000 differences between you and I just in our gene sequences, and they only represent 2% of the human genome. When we go on to genome sequencing, which is sequencing the entire human genome in an individual, we've got 3 billion nucleotides to read. And this is very challenging because then we're looking at about 5 million differences or 5 million variations in the sequence between you and I. So that's the challenge. The major challenge we have is, of course, we have solved an awful lot of these cases, and we've got over 270 genes that we already know are implicated as the cause of inherited retinal dystrophies. Nevertheless, when patients come into clinic and we do diagnostic testing or research, we're still left with about 40% of cases that we cannot solve very easily at all. So we really have a challenge here. And the way we've addressed this challenge is to collaborate with all our colleagues across the UK, our clinical colleagues, our scientists, to speed up this discovery so that we can share data. So this is called the UK Inherited Retinal Dystrophy Consortium, and this is funded by Retina UK. And what we're trying to do here is have a collaborative approach to understand inherited retinal diseases with the aim of accelerating the discovery of genes, improving diagnosis, and developing the data and sharing the data so that we have the capacity to learn from one another. And this was established initially through 4 centres in the UK, the University of Leeds, led by Chris Inglehurn, the University of Manchester, led by Graham Black, and the University of Oxford, led by Susie Downs, and of course our grouping at UCL Institute of Ophthalmology and Moorfields Eye Hospital. And this has just been an absolutely wonderful, wonderful experience, bringing people together and making really significant discoveries. So as I said, we've got clinicians and we've got scientists, but we also have bioinformaticians. And if you haven't heard of that word before, these guys are the computer wizards. You can imagine that, you know, with billions and billions of letters to read, it takes a lot of computer power and a lot of computer programming to help us to read the data so that we can try and interpret what's going on in terms of variation in the human genome. So we are selecting in this consortium individuals, patients and their families who we can't solve, who've been very, very difficult to solve over the years, using our current pipelines of diagnostics and research. And we've sequenced about 500 individuals, and we have another about 100 individuals ready to go on the machine. I think they're running right now, actually. And what this has enabled us to do is create an efficient infrastructure where we can share the clinical information and the genetic data and all our combined knowledge and expertise, so we can have really informed discussions and try and interpret what's going on in the genomes. This has been very, very successful. We've discovered many new genes, different variants that we weren't expecting, and also unexpected association of a gene with a phenotype. So this goes directly back into the clinic and the diagnostic pipelines. So many, many families now have a genetic diagnosis by coming through this UK Inherited Retinal Dystrophy Consortium, who otherwise would never have been solved, and they probably would never have been studied. So it's an incredibly valuable approach to have a consortium around the UK that can then inform diagnostics. So that's some background for you. What I want to do now is to give you an example of a couple of the new discoveries in my lab. And really what I'm going to describe to you in the rest of the talk is how we've utilized these technological advances in next generation sequencing and in stem cell biology to understand the genetic variants that cause retinal diseases and to test potential therapies, which is very exciting. So this is one of my favorite genes that I've worked on for many years. This gene causes X-linked retinitis pigmentosa, and it's called RP2. It accounts for about, variants in this gene account for about 15% of all the X-linked retinitis pigmentosa patients that we see. And it's an interesting gene. As you can see from the diagram here, these blocks, these are the exons in the gene. It's quite a small gene. It has 5 exons. Some genes have up to 100 and more. So this is a relatively small gene. We've just finished a study looking at the individuals who attend Moorfields Eye Hospital, and these are the changes that we see in their RP2 gene. And you may not be able to realize this from the diagram, but stop mutations are very common in the RP2 gene as a cause of X-linked retinitis pigmentosa. So that's a stop where we change one letter and it stops the genetic code. So we don't have a sentence. So in other words, we can't make a protein. So this is something we've been interested in for a long time. But we've harnessed new technology. We were one of the first to do these types of experiments. I have to say it's really, really exciting to be in the lab in such a time where we've got technological advances that allow us to do research that we could never dream of doing before. So what we've been doing is taking patient stem cells. And how we do this is we take a little biopsy from a skin sample, and we put them in a dish in the lab and we grow out those cells. So we basically grow out the skin cells. Then what we can do is we can reprogram those skin cells. So here's the skin cells in a dish here, if you can see that. We can reprogram them and make stem cells, which are pluripotent. So here's an example of a stem cell colony. Now these stem cells are pluripotent, which means in theory, we can change them into any cell in the body by differentiating them and looking after them in a very particular way. And what we're able to do is we're able to make these stem cells into retinal pigment epithelial cells. Excuse me for getting a bit tongue twisted there. So these RPE cells are actually the supporting cells for the photoreceptors. They look after the photoreceptors, they provide them with nutrients, and they take away anything that needs degrading. These RPE cells are very, very important in a human retina. And then we can purify these colonies and make colonies. So it's, in a sense, what we've got is a stem cell derived RPE culture from a patient with their mutation or their change in their DNA in a specific gene. And that then enables us to do experiments in a dish in the lab on these cells, which are human cells that we've never been able to do before. So here's an example of RP2 with a full stop mutation. And you can see here's a control and here's the patient cells, and they make nice RPE cells. So that's already telling us that if you have this change in RP2, you can make RPE cells. And then we can do lots of fancy experiments like these microscopy images shown here to see if there is anything wrong with these RPE cells at all. But what I wanted to share with you today is the way we tested a potential therapy. So I think you may remember that I said stop mutations or full stops are very common in RP2. So what we thought was we could potentially try and use a drug that ignores full stops and see if that helps put RP2 back into these cells. So this is exactly what we did using these stem cell cultures. We used a drug called PTC124. This drug is in late clinical stage development for Duchenne muscular dystrophy and cystic fibrosis, because they also have full stop mutations in those genes. The diagrams on the right you don't need to understand, but that's just to show you and to help me clarify that actually when we add this drug to the cells have no RP2 at all because they can't make the sentence, they can't make the protein. But when we add the drug, we see the gene is made and there's a little bit of protein made. And we've done some more detailed experiments to show that little bit of protein actually restores function in these cells. So this is really exciting. So we've shown that we can use a drug, a read-through drug, we call it, to restore RP2 gene and protein expression in eye cells in a dish as a potential therapeutic for this condition with that particular type of variation in the gene. But what's particularly exciting is actually our ability to make a retina in a dish. So this is a little bit more complicated than the previous example. It's the same principles. What we do is we take a skin biopsy, grow it in a dish, and here you can see the skin cells growing around here. Again, we reprogram them and make stem cells. But instead of making RPE cells, this time what we're trying to do is grow a whole neural retina with all the different neuronal layers and cell types in that neural retina, including rod photoreceptors and cone photoreceptors. And this is what it looks like in the lab. And it's amazing to see. It's just so amazing. It's a beautiful thing. And the technological advance here is just astounding. Here you can see it growing. And here, hopefully you can see that there's this little clear layer at the edge of this patch of cells here. This is the retina growing in a dish. And this is actually what it looks like at the end of the protocol that we follow in our experiments in the lab. So here we have a retina in a dish. Right, now the possibilities are amazing of what we can do with this. I just want to show you some details of it, some really close-up images using microscopy. Hopefully, when we look closely at these retinal organoids or these retinas in a dish, what you can see here is all the different cell types that you would expect to see in a human retina are there. We can see rod photoreceptors, we can see cone photoreceptors, the blue ones, the green ones, and the red ones. And we can see what we're calling here rudimentary outer segments. So this is the light sensing part of a rod photoreceptor and a cone photoreceptor cell. So this is just amazing. It's beautiful. It looks just as we'd expect it to do. It follows human development perfectly. So the cells grow in a sequence of events as we'd expect to see normally in a human. Now these, of course, are tricky experiments. It takes you more than 6 months to grow a retinal organoid, a tiny little retinal organoid, and they need lots and lots of attention and care while we're growing them. But we now have a whole group of experts in the lab who can do this pretty routinely. I shouldn't say that, fingers crossed. So I'm going to talk a little bit more about what we can do with the organoids, but I thought it was important to also mention something that you would probably have heard about because it got the Nobel Prize, and that's something called gene editing using CRISPR. And what we can do now is we can gene edit. So when we've got the stem cells, we can actually gene edit the DNA and make knockouts or mutants, or actually try and cure the mistake by correcting it. So this is proving to be an incredible, the limit here is just your imagination in theory of what we can do. So I just wanted to show you an example of how we've used CRISPR. We have actually CRISPR corrected RP2, but I don't have that data on these slides for you. Here we've used it to create another model by knocking it out. So not only do we have the patient cells where they have a full stop in the sentence, we also have a gene-edited CRISPR knockout cells. And as you can see here, in a normal cell, we've got RP2, and in the CRISPR gene-edited knockouts, we have no RP2. So it's like the patient cells. Here you can see when we look in these organoids of the patients and the CRISPR-edited cells. So here we're looking at the retinal organoids, and the green staining here is RP2, and this is in a normal retinal organoid. When we look in the patient cells or the CRISPR knockout, there's no green. It cannot make RP2 because it cannot read the genetic code. So this is wonderful because now, for the first time, we can look at a human retina in a dish and study what's going wrong, and then hopefully come up with some therapies. So the question we have, if there's no RP2 in the retina, how does it cause the retina to degenerate? And now hopefully you can appreciate on this part of the slide on the right-hand side that in the control organoids, we have lots and lots of healthy cells with RP2. In the knockout, or the retinal organoids lacking RP2, the retina started to degenerate in a dish. So you can see the number of cells is reducing and reducing. So we have only limited number left. So we can actually now model disease in a dish. We went a step further because not only can we model the disease in a dish, but we can also test therapies. So similar to the read-through therapy that we used before, this time what we tried in the retinal organoids was actually gene therapy. So you can see in the retinal organoid on the left, this is a gene-edited CRISPR knockout RP2 organoid. And you can see there's no green here at all because it doesn't have RP2. We introduce a gene therapy vector containing RP2 gene, and we put it on the organoids, and hopefully you can really appreciate the fact that we've got loads and loads and loads of green here. It's just amazing. What this is showing us that RP2 can transduce the retina. It can get into these photoreceptor cells and restore RP2 protein in these cells. So this is an example of how we can test a gene therapy in these retinas in a dish model. And the beauty of this is that actually gene therapies have traditionally been tested on mice or dogs, and actually mice and dogs are different. And we've never really been able to test it on human cells like this. So this has really revolutionized how we can approach a gene therapy approach, because we can now see if the gene therapy construct is getting into the right cells and how much of it is getting into the right cells. And we can tweak it and do different experiments to maximize the opportunity of restoring function to the retina. The great thing is that not only do we get lots of RP2, we can see that rhodopsin is expressed in the gene therapy organoids, and also that means they're making rod photoreceptors properly. When we looked, remember earlier I said what we noticed is the retina starts to degenerate when you don't have RP2. But after we've done gene therapy in these retinal organoids, we can see that actually the retina gets thicker again. So it can actually restore the retinal thickness and help those cells to survive and prevent degeneration. So we're very, very excited about this work. This really shows the utility of using gene therapy as well as an approach for RP2. And obviously we're doing this type of work for many, many other conditions in our lab in collaboration with Professor Mike Cheetham, who's our expert in retinal organoids. The gene therapy angle is from Jane Farrer's group. She made the virus containing the RP2 gene for us to test in these organoids, and it's a very, very good virus. It works beautifully. So that's Jane Farrer's group in Dublin, who contributed significantly to this translational research. So the second half of my talk, I'm going to talk about, so hopefully I've shown you how retinal organoids and stem cells can really help us advance our understanding of disease in a dish and develop therapies. Now what I want to tell you is a little bit more about how we can discover new things in genetics and genomics for inherited retinal dystrophies. And again, we use retinal organoids for this, but I'll come on to that. So here's an example of a huge pedigree that was actually drawn up at Moorfields Eye Hospital over 40 years ago. This family suffers from dominant retinitis pigmentosa, and it's remained completely unsolved for all these years. So we took the challenge and we decided to genome sequence, so sequence the whole 3 billion nucleotides in this family through the UK Inherited Retinal Disease Consortium. Many thanks to Retina UK for funding this research, of course, without which it wouldn't be possible. And to cut a very, very long story short, with lots of computational biology, genetics experts in the lab, and lots of hard thinking, what we found, and these are very difficult to find because these families remain unsolved. Remember, I said 40% of families remain unsolved. They remain unsolved for a reason, and that is because it's very, very difficult for us to find these changes. They're more complex than the obvious changes we're used to finding in previous years. So what did we find in this family? We found a structural rearrangement of a chromosome. And what I mean by that is there were multiple copies of chunks of DNA, and some of them weren't just more than one copy, but they were also the wrong way around. So they were inverted. So this was very exciting. And then we went back and looked in all our data from across the UK, in our UK IRDC consortium, and also in Genomics England, the 100,000 genomes effort that's gone on. So there's lots of information in there that we can tap into and explore. We found that there were another 11 families in the United Kingdom who had exactly the same structural variant as the first big pedigree that we were looking at from Moorfields Eye Hospital. So this indicates that there is a founder effect. This change has been around for a while in our population. Of course, then we were thinking, well, if these families have a structural variant, are there any others that have a structural variant in exactly the same place? And where we're looking now is on a chromosome called chromosome 17. And we found 3 other families in the UK, and these 3 families all have different structural variants. So now we have 4 different structural variants in the UK associated with dominant retinitis pigmentosa. And we were very fortunate to work with a wonderful group in the Netherlands, Suzanne Roosing's group, who were equally confounded by a family they had, the top family here from the Netherlands, a huge family with dominant RP. They couldn't find a change in a gene either. And they had a structural variant in their family in exactly the same spot as ours, but a different one. So we worked really closely with Suzanne and her amazing group in the Netherlands, and we were looking at families from all over the world. There is a South African founder structural variant, and these are the South African pedigrees here. You can see there's 4 pedigrees here. They all have the same structural variant, which is different from all of the others we found. Here's a large Canadian family, and Suzanne also found another structural variant in another family from the Netherlands. So there are lots of other families out there with structural variants on chromosome 17. So this is what we call RP17. So we found these structural variants, and we were completely convinced that they must be the genomic cause of disease. But how? Because we're not really looking at a gene here. We're looking at something in 3 dimensions, okay? So this was a challenge for us. So we've collaborated with some experts in Germany who use a new technique, and I won't go into the details, but it's a brand new cutting edge technique where we can look at the 3-dimensional structure of the DNA on that chromosome. And this is what the data look like. Difficult to interpret if you're not an expert. This is the first time this experiment has ever been performed in human retinal tissue. And of course, what we did was we grew our beautiful retinal organoids from a control individual and from the individuals with these structural variants with RP17, and we did this experiment. And you can see there are differences. Hopefully you can appreciate that the two pictures here aren't identical. And what did it mean? So then, again, to cut a very, very long story short, essentially what we have here is the wrong structure of the chromosome. So in a control, we can see a big loop of DNA here with an on switch. This is an on switch for retina, and this switches this gene in this loop of DNA on in the retina. That's a normal situation. In our RP17 individuals, what we have is, because we have these structural rearrangements where we have extra bits of DNA all around this region, what all of these 8 different structural variants had in common was this. that actually we had an extra copy of all of this that was a little bit wrong. And essentially what happens is we have another on switch here that also then, whoops, apologies, just go back one, switches on this gene called GDPD1. So this is our model for what's causing RP17. Essentially, the altered chromosome structure enables an on switch that switches things on in the retina to have contact with a gene that it shouldn't have contact with. So in other words, it's asking that gene to switch on in the retina when it shouldn't, and therefore we believe that is toxic. And that's the next phase of our research, to really prove that this is the mechanism of disease. So far, and really we've only just begun looking. I can tell you now we already have another structural variant identified. We have at least 9 structural variants in more than 22 families with dominant RP, with over 330 affected individuals. So this is actually quite a common cause of dominant RP, and we're only scratching the surface. We've still got a lot of patients and samples to look at to get to the bottom of how common this really is. So hopefully you can appreciate that we've discovered a major cause of dominant RP, and that now enables us to do diagnostic tests in those families and in unsolved individuals. What's interesting is there was an incorrect gene that was previously assigned as causing this form of RP on chromosome 17, and that is now incorrect. So now we have a correct cause of this condition. Not only that, we've discovered a whole new mechanism of disease, this altered 3-dimensional chromosome structure and DNA structure. So now we can appreciate that we might not just be looking for genes. What we're looking for is all kinds of changes in the DNA, including all that DNA that doesn't code for a gene, and what the impact of that is on the retina. So now we know that we've got this new mechanism, we can go in and find more similar types of mechanism, and we're highly likely to find more of these. I also hope you realise that making retinal organoids, although it's challenging and takes like a year from stem cells, it allows us to investigate these genomic changes in human retinal cells for the first time. And this is a huge revolution in our field, and it opens up so many possibilities. It gives us new understanding that will help us in all our future genetic and functional studies. And hopefully I've managed to demonstrate to you today as well that it will help us to develop and test therapies for inherited retinal dystrophies. So thank you very much for your attention. Obviously this science isn't done in isolation, it's a huge collaboration. I mentioned the UK IRDC, which is centred in London, Leeds, Manchester and Oxford, but we also partner with Southampton, Cardiff, Glasgow, Bristol. This is the team in the lab who do the work. So Mike Cheetham is our retinal organoid guru. There's a whole host of postdocs, PhD students have helped with all this work. Alessia Fiorentino, who was in the UK IRDC, really led on the RP17 story. Our wonderful colleagues in the Netherlands, who found all those other lovely structural variants as well. Our colleagues in Germany, who helped us with the 3-dimensional sequencing. Of course, our colleagues at Moorfields and all our other centres around the UK and elsewhere. And Jane Farrer's lab, who we worked with on the gene therapies. And of course, huge thank you to our funders, especially Retina UK, who's funded all the UK RIDC work alongside Fight for Sight. And just to finish, here's a picture of the main entrance of the Institute of Ophthalmology, which I've been missing lately, and some of the team involved in the work. So thank you very much for your patience and for listening today, and I look forward to having conversations around this with you.